And welcome everyone. My name is Raeka Aiyar, and I'm the Vice President of Scientific Outreach at the New York Stem Cell Foundation Research Institute, or NYSCF as we call it. I also oversee NYSCF's diversity, equity, inclusion, and belonging efforts, and today's conversation will delve into a critical part of these efforts. So thank you all so much for joining us. NYSCF's mission, as many of you know, is to accelerate treatments and cures for the major diseases of our time. That includes ensuring that these treatments can benefit each and every patient in need. As we all know, diseases affect people differently and drugs affect people differently. That means that a one-size-fits-all solution, and more importantly, one-size-fits-all research, is not going to give us the answers that we need. The only way that we can account for these differences and deliver on the promise of precision medicine is to ensure that our disease research and our clinical trials are inclusive of all populations impacted by disease. This sounds obvious, but the reality is that this has not been and is not the case. Even though the mortality rate for major diseases like heart disease, stroke, and cancer has decreased, thanks to many breakthroughs going on in the community, the gap in mortality between white patients and patients of color has been increasing. For example, a Black woman is 22% more likely to die from heart disease than a white woman, and 71% more likely to die from cervical cancer. Before COVID, less than 5% of NIH-funded respiratory research reported inclusion of racial or ethnic minorities. All of the diseases that we work on at NYSCF, and probably what many of the scientists listening today work on, have disproportionate impacts on ethnic minorities. This is an unacceptable state of affairs, and there are many reasons for it, all of which are rooted in systemic racism. And we will touch on these reasons today. But our focus will be where we can make an impact in the realm of disease research. Tackling these racial health disparities is mission critical at NYSCF. Due to these kinds of systemic discrimination and racism, many minoritized populations have been excluded from research, and as a result, we have a biased understanding of disease and treatments that do not work for everyone who needs them. These issues of diversity, equity, and inclusion are incredibly close to our hearts at NYSCF and have really been embedded in our DNA since our founder and CEO, Susan Solomon, started NYSCF. One of Susan's earliest priorities at the Research Institute was to create the NYSCF Global Stem Cell Array, our technology for automating stem cell production from hundreds of patients at a time, to ensure that we could represent the diversity of the world's population in stem cell research. Thanks to this technology and to many other excellent efforts in the community, including by our esteemed panelists today, we now have an opportunity to rectify the biases in disease research and move towards inclusive precision medicine. This is a really important conversation, and we all need to work together on this challenge if we are to move forward. So this is also a call to action for everyone in the audience today. Before we begin, I would also like to acknowledge the transformative partnership of the Stavros Niarchos Foundation in making events like today's possible and encouraging others to support all of our efforts. To all of our visionary partners who are watching today, we are deeply grateful to you. Our research and discoveries continue to be fueled by your philanthropic support. For those of you who are new to NYSCF, we look forward to sharing more of our work with you and hope that you will join us in our mission of accelerating cures for patients everywhere. We will be incorporating audience questions as we go along and encourage you to use the Q&A feature in Zoom to submit those. So I am honored and privileged to be moderating this panel today on such a critical topic. And we have a really outstanding lineup of panelists with great perspective on the unique challenges around diversity and equity in biomedical research and clinical care. And I'll invite our panelists to join us now. We're really glad today to have Ekemini A, the founder and president of the Coalition for Aligning Science, a boutique scientific advisory group that integrates biomedical research, philanthropy, and social impact. Dr. Greally also serves as managing director of Aligning Science Across Parkinson's, a research funding initiative for Parkinson's disease. Thank you for being here, Ekemini. We are also happy to have Ru Gunawardane, the executive director of the Allen Institute for Cell Science, a cellular biologist with a great track record in drug discovery in the biotech sector as well. Ru, thank you so much for being with us today. Our third panelist is John Greally, a professor of genetics, medicine, pediatrics, and director of the Center for Epigenomics at the Albert Einstein College of Medicine. also a clinical geneticist and active clinician at the Montefiore Medical Center, one of the top hospitals nationally and a leader in LGBTQ care, serving Bronx as one of the most diverse counties in America. So John, thank you for being with us today. And last but certainly not least, we have Scott Noggle, NYSCF's Senior Vice President of Research, who spearheaded the stem cell technologies at NYSCF, including the NYSCF Global Stem Cell Array, that allow us to study disease at a population scale, which is critical for taking on the issues that we will discuss today. Scott, thanks for being with us. So let's dive right in. The question I want to address first is, why is it so important for disease research to represent diverse populations? We know that there are deadly consequences of this. What is happening because we've missed this? Just last week, Bloomberg highlighted the startling exclusion of Black patients in clinical trials for Alzheimer's disease, a population that is more prone than white people to develop Alzheimer's, but only represent 2% of trial participants. John, let's start with you. As a clinician and geneticist, you personally witnessed the impact of these types of disparities on patients. Could you provide some examples of how this shows up and treatments that might be ineffective or less effective in minoritized populations?
Thanks, Raeka, and thank you for the opportunity to participate today. It's a really important topic, and I'm surrounded by great colleagues here. I'm looking forward to this. I'd like to make the point at the outset that, and this is something we're very sensitive to up here in the Bronx, that health disparities are not based on our genomes. Health disparities are almost overwhelmingly based on the socioeconomic policies, the biases, the racism that exists in society. What we can and should do in terms of research is to try to bring the same amount of genomic insights to all populations so that we can provide the same care to everybody, no matter what their ancestry should be. And that's something that we have an especial challenge in doing in the Bronx, where 91% of our population is other than Northern European white. So some of the things that we're particularly interested in the Bronx, we always talk in the Bronx about ABCD being the priorities in terms of healthcare: asthma, blood pressure, cancer, and diabetes. And asthma in the Bronx, it's a very polluted place with very large highways going through. What we know is that the treatment of asthma using the standard bronchodilators that relax the muscles of the airway and prevent spasm, that they work much better in some ancestries than others. So for example, if you have, there's a study that I'm just looking at the numbers from here. The non-responsiveness to albuterol, which is our first-line treatment, is about 47% of people who identify as African-American and 67% of Puerto Rican kids. Work by Esteban Burchard and UCSF will point out that when you then look at Mexican-American kids, they have a much better response to the same drug. So this is and as background, it's about 20% of people who are Northern European white who would have a poor response to it. So we have multiples of this difference that exists between these different groups. The thing that this really highlights to me, among other things, is that you have this useless designation of Hispanic, within which we have Mexican Americans and Puerto Rican Americans with widely divergent differences in terms of how they would respond to albuterol. Which is it just shows you how limited our the information is that we collect in terms of ancestry to allow precision treatment. That's at the whole population, sort of race and ethnicity level. The one other thing that I would add in, which is a great example that my population genetics colleague Sri Raj here at Einstein has told me about. There's a group of people in India called the Vizia. And they're quite an endogamous group. They've been they have their own sort of genetic pool in a similar way to the Finnish and the Irish and other populations. But they have about 100-fold higher incidence of a mutation or variant that in a gene that is required to break down certain medications that we use when we're about to intubate a patient for surgery, with the result that these drugs that we're using to relax the muscles will be prolonged in terms of their activity in people from this particular ethnic group. So this is an example of going from the very broad categories to something very, very specific, and having a drug which is contraindicated if you go into the local hospital and say, this is my ethnicity, don't put people on this drug because they may die. So we have everything in between, broad racial and ethnic groups down to very specific subpopulations. And unless we study them, we're not going to understand where we could be doing harm with these sorts of medications.
Thank you, John, for starting us off with some really, really great points. And we'll definitely come back to the socioeconomic implications shortly. But, you know, I mean, you talk about understanding the kind of genetics of what's going on here. So that points to another issue, that even our basic understanding of biology and how cells work and how health works is itself biased, since we've only, you know, primarily been looking at these white European populations. Ru, this is something that I know is really of interest to you at the Allen Institute, and you recently co-authored a really great piece with Ekemini in Nature Cell Biology about the issues in this. Can you speak more about how this radiates across the research pipeline, even to drug discovery, which you know, you also have experience in?
Yeah, thank you, Raeka. And again, thank you, and to all the panelists for starting this very important conversation that could take days to really cover appropriately. So yeah, so I'll start with the basic research piece, because that's what I'm doing right now, or the group at the Allen Institute for Cell Science. We are trying to understand how a normal cell works with imaging, gene editing, all the cutting edge technologies we have right now. But I think we can look around and say, how do you define normal? Right? And I think we really need to define that better before we even can think of what abnormal or pathological really means for drugs, for therapeutics, and really for truly personalized medicines. So then that's where stem cells come into play. Our model system is stem cells because they are more normal than a lot of the cells we have used in basic research in the past. Not just because of lack of diversity, but because they're genomically unstable. They allow us to work in the research lab. So, but the problem right now is, even if you think of some of these tools that we use for understanding what normal is, it doesn't capture the actual diversity in the population. For example, the reference sequence we use, right? There's one ref-seq that we compare all of our...
Could you define what you mean by ref-seq?
Yeah, the reference sequence of a human genome that we compare as we do kind of quality control experiments, as we make new cell lines for research. And so that's our definition of what normal is. And if in fact we look at cells through the microscope, right, and we are looking at the subcellular organization of cells, no two cells look alike. And that's from one clonal cell line. But what about if we can capture cell lines from different backgrounds, different ethnicities? How wide will that spectrum be of what we define normal? So I think this foundational knowledge, from basic research to building model systems where we can introduce mutations, introduce drugs, that's, I think, what's missing right now. And that's going to help us not just broaden the scope of, you know, those kind of model systems, but also give us that understanding of how human biology works at the smallest unit, at the cellular level. So that's what's really exciting, and I think is very much needed, rather than working on one cell line and, you know, defining what normal is. Now, to your question around how this applies to looking for new therapeutics or drug discovery, a lot of the initial screens that happen in a drug development program uses non-human cells because they grow and they're easy to work with. But when you get closer and closer to human cells, like primary cells, and now more recently stem cells, again, it's more about how reproducible that cell line, how robust that cell line is. We don't really think of diversity as a criteria when we pick those cell lines. that define the experiments, that define the assays, that define the criteria before they move on down the drug discovery pipeline, right? So if there are pathways like John was talking about that are affected in a specific population, that's just not even in our scope of work. We don't even know there are differences there. So there may be pathways affected or not affected even in the initial screening process because of the cell lines or the reagents or the biological tools we use, because it's very narrow right now.
Mm-hmm.
Yeah, so
Absolutely.
to get towards personalized medicine, I think it has to start from the basic foundation and work our way up all the way to testing drugs in humans.
Right, absolutely. Really great points. A lot of missing biology out there that we need even just as a baseline to begin to ask these questions. Ekemini, I wanted to go to you next because you co-authored this really beautiful piece with Ru recently. Can you share with us your perspective as a molecular biologist, as a funder, as a policy advisor on these opportunities before us?
Sure. I mean, well, that's pretty broad, but I would say that, you know, as I'm hearing John and Ru speak, I think it's very clear that we have made some strides at the population level, right? I mean, even with the advent of the All of Us study out of NIH and a few other smaller studies that are looking at diverse groups, we are wholly missing that at the basic science aspect. So I think, I mean, we all know that the healthcare all the way to science pipeline is much broader than workforce, than our clinical coordinators. And I think we need all of it, everything from clinical all the way down to basic, to really be able to actualize personalized medicine. So I know later in the conversation, we'll get into really deep topics like polygenic risk scores and how you're getting genome-wide association studies and how that's, you know, being used at the research level, how people want to use it clinically. And I think there should be a lot of caution taken before we're at the place of using these things clinically if we know that we don't have representation across the board for diverse groups. We are already running into issues clinically, as John has described. And I think, you know, once we start getting to the place where we're refining what we're doing genetically, using that clinically speaking, I think we can get ourselves into a lot of problems without really truly understanding and incorporating diversity early on. So I'll say there that from my perspective as a funder, there are a lot of, you mentioned at the top, we're, you know, also talking about solutions here. There are a lot of things I think that funders can do to make sure that we are pushing the envelope, which I know we'll get into a little bit later.
Thank you so much, Ekemini.
Of course.
When we're talking about this missing biology, I mean, we're talking about the value of stem cell models for capturing some of it, at least when it comes to the research aspect of this. So Scott, since you lead our research efforts to do this here at the NYSCF Research Institute, can you speak to some examples of how we're taking that on and how that has yielded some of this missing biology that we didn't have using kind of the standard approaches?
Yeah. So you mentioned earlier, you know, I guess more than a decade ago now, we started trying to build the infrastructure that would enable us to scale up these studies of individual cell lines to be able to look across, you know, populations. That was sort of the original vision of creating all of the automation and you know, all the infrastructure that we put in place was to be able to look not just at one or two individual cell lines at a time, like Ru said, but really be able to look at, you know, larger populations in parallel. So, you know, taking this idea of using a stem cell as a model for a disease, you know, being able to turn that stem cell into the various cell types that we think are affected by disease, and then looking across, you know, as many individuals as we can, you know, in one go. The reason for doing all that, you know, and putting that infrastructure into place was to try to minimize, you know, unwanted variation that we know arises during, you know, running routine experiments. You know, one of our goals was to try to minimize all of that sort of experimental noise and get down into, what is actually represented by the biology of the diseases that we're looking at. So, we're doing this in multiple different disease areas. I think one example that we've just published on recently is a study that we've done with David Bennett at Rush University, who oversees the ROS-MAP cohorts and studies, and Tracy Young-Pierce at Brigham and Women's, looking at Alzheimer's disease and seeing if we can faithfully represent Alzheimer's disease, particularly this late onset form of the disease that we're trying to understand, in stem cell models. And we were only able to see differences between AD and controls when we looked at a large enough population. And there wasn't any one particular AD or one particular healthy control cell line that really gave us a nice view between healthy and disease when we looked in the model systems in vitro. We really needed to have that sort of larger spread of multiple examples of healthy and multiple examples of disease to be able to tease apart what that disease actually looks like when we look at it in the dish, and then be able to compare that back to sort of in vivo data that we had gathered at the same time.
Data from the patient's brain.
Data from the patient's brains in particular. So and we found that we could find that correlation, but it was spread across the entire population. So we need, we're, in part of these studies, looking at the genetics and how that correlates to the phenotypes that we see in vitro. As Ru said, that there's lots of biology that we think we can capture looking at population. But we know that the particular set of cell lines that we looked at in that study were largely from backgrounds of white Northern European ancestry. And so there's a real fear that we're missing a part of the biology that exists, that's important, not just in Northern European populations, but on all the other populations as well. And so we're actively aiming to expand the diversity of those of those cohorts, of those studies, and be able to include more diversity into those studies. But I think there's a long way to go to actually realizing that, to be able to understand how variation impacts the biology that we can see in vitro, to really have a faithful representation of the disease across the entire population.
Absolutely. Well, we know, I think now, why this is an important issue, why we want to increase the representation. So why don't we talk now about the current state of stem cell biobanks and disease research, the factors behind the lack of diversity, and start to talk about how we can overcome these. So when we talk about disease research, for example, 78% of people in genetic studies of disease up to 2018 were of European descent. And when we talk about stem cell biobanks, the majority that are out there have similar proportions. And there's, of course, many reasons for this. And as John rightly pointed out, there's definitely a social and cultural aspect to this, and systemic racism at the root of all of these. And we could spend a lot of time talking about this. But I think firstly, we need to call out the elephant in the room, that a lot of these communities rightfully have a mistrust of biomedical research due to a lot of atrocities and mistreatment by the medical community, ranging from the Tuskegee syphilis trials to the legacy of Henrietta Lacks and more. So, and this was also raised in an audience question. So what are the fears in minoritized communities around participating in research? And how do we effectively account for these and work to build trust back?
I can kick off here because I think there are a number of different reasons. And through my work with the Global Genetics Parkinson's Program, or GP2, that ASAP funds, what we are seeing is that their various communities don't necessarily want investigators to come in, take samples, and then you never hear from them again. I think what we are learning is that there needs to be a give and take, right? I mean, we know that we need samples for research. We know there's a lot to be gleaned from human material, but then, what do those communities receive back? Do they receive back return of results when there's genetic sequencing being done? And how are the benefits of research, or what is being sought after, really being explained to those communities? I think in that, we need to think about how we are engaging with various communities, how you are working with community leaders to actually reach out to those in the community, and that you're not making empty promises, right? So I think I've seen efforts that make things, in my view, far more difficult than they need to be. People of all different backgrounds. I mean, if you take the time to explain to them why it's being done, what's the purpose, and what they'll be getting from it, and what their data or their material will be doing for research, I think, starting off at that level would be fantastic for all groups involved. So I think that's one big issue that I've seen, is that you're making sure you're not going into communities and swooping in and swooping out without ever communicating, what is being done and how they can benefit, as well as the research community.
Right, it's a critical part of the process. John, as a clinician and someone who works with patients directly, what would you want to add to that?
I feel very awkward as a senior white guy talking about this stuff, to be honest with you. It becomes a they issue. I don't want to talk about they. I want to talk about an inclusive us. But I'm not the person to go out and speak to the people in the Bronx, despite the fact that I would have all the goodwill in the world to try to help my patient population. So we've been thinking a lot about this at Montefiore and Einstein, and our approach is simple. We're putting together 5 principles of how we should be doing genomics with our community, not on our community. It's going to be with and for. And people like myself and my colleague Sri Rajesh, who I think is one of the participants today, we're going to sit back and we're not going to lead these efforts. We're going to have our population health colleagues talk to community representatives, and they will work out what they want between them. And then they will come to us, and we're the technicians who will implement it and try to deliver it back into the electronic health record system within our health system. The community outreach that I've been doing so far has been really interesting to me. And it speaks to one of the issues that Ekemini raised and that you have raised as well, Raeka, this issue of mistrust and having a meaningful partnership. I had pushback from a Black woman from the Bronx who said, when you want to do this genomics on our communities, you are targeting us, right? And I said, yes, we are, because we are. And that information could be used to kill us. Now, at one end of the spectrum of possibility, you could potentially make that argument. I was not going to enter into this discussion because it's not my place to try to unravel hundreds of years of racism that led to that statement. But what it does do is it says that we have to have very clear guidelines for how we would set up these interactions between our medical center, our medical school, and the community that surrounds us completely here in the Bronx, so that people understand that this is not a risk. And the only way of making sure that it's not going to be a risk is if the people of the Bronx own the process. And we have to get away from some of our traditional models for research, in which it's people like me with a big NIH grant who ultimately makes decisions about people's data. That's not going to work here.
Really great point. And that points to the, sort of how the standard research recruitment pipeline works. Certainly there's room for improvement there, and that's part of the reason that we're not seeing the extent of diversity that is really needed. So what are some ways that we can change how we recruit? How do we better integrate the voice of the patients, the voice of the communities that we need? Scott, I know you've been thinking about this a lot in terms of the work with Alzheimer's that you mentioned. Anything you'd like to add there?
Yeah, I think just building transparency into the research programs that we do, being able to put into place programs to to communicate the results that we do find, that we are making step-by-step progress towards the goals that we have. Having that be a part of the program as a whole, and the research that we do, not just as a side thing, but completely integrated into the way that we build our resources, build our biobanks that we use to understand the disease process, from a biological point of view. And I think the other thing is important is to try to eliminate barriers to participation. There's often a desire to try to build our cohorts using very strictly defined criteria, and a lot of those criteria can be basically cut off the source before you even have a chance to interact and have those people participate in the programs. So we've gone back and really seriously looked at the exclusion criteria for how we enter people into the research studies that we do, to make sure, at least try to eliminate some of the bias in some of those exclusion criteria that we might have artificially thought we needed, but probably don't need, and it's probably inhibiting our ability to understand disease across the population. So I think it's having an active program to try to eliminate these in a more active sense is really one of the main ways we can probably change how we're approaching some of these studies and building these biobanks.
Yeah, really great point. And this is, I think one aspect is when it's patients and they maybe more intrinsically have a motivation to participate in research because they're hopeful that it could benefit them. But we're also, some of the work that we've been doing with John's group at Einstein is to even just understand this baseline biology, as it occurs across different populations. And these are from healthy people who hopefully have no diseases, but just to get that at a baseline. Ru, since this is an interest of yours, how are you thinking about this and getting people in the door to be able to represent that in an inclusive and in a respectful way that acknowledges these fears?
Can you state the question again? So I was going to say it's around education and sharing, and that the part you would like me to speak on.
Just how you're thinking about it at the Allen Institute in terms of bringing in broader populations while factoring in these hesitations.
I think from our point of view, since we are again studying more foundational biology, and as our founder like to say, decoding the cell, how we go from all these genes to how the cells behave and function to how the cells go wrong. So our approach is through sharing. I think communication is a good word here. It's a broad sense of communication, sharing our tools, sharing how we do this very openly, not always waiting for publications, official publications that take a very long time to get out there. And they are more for a kind of a very specific, specialized scientific audience, sharing stories about how what we're doing impact the science, impact the knowledge. because we are a nonprofit research organization. And then also doing kind of education-wise, reaching out to more educators on how to talk about what's happening inside our bodies, how do we figure out what's happening, the resources we use, the imaging, the gene editing we do so we can look inside the cell so that people are curious, right? And I think if you can get people curious, then that's a way to communicate. It's not, you give me this so I can do my research. It's like, look what we are learning. Look what stem cells have allowed us to do. We can study heart cells now. Maybe then we can study how it goes wrong. Maybe then we can figure out how to correct that. Because the amazing thing about stem cells here is you can make different types of cells. And those are things like looking at a beating heart cell is exciting even for me, right? So it's the educational open sharing aspect on the broader level. And I think there's another part to this. I think this kind of work is not easily funded, right? So another aspect is to make this data, the tools we generate, available to the research community at large, and to do that in a standardized way so that experiments done in a lab in China can be shared with the lab experiments done in New York, right? So we spend a lot of time, and I know you do too, at NYSCF, as you call it, that's new to me, standardization, right? So that the quality of the tools are standardized, they're reproducible, so that others can use it. And the same thing with the data, so others can look at it and see what they can gain for their own research. So there are two aspects, one for the people doing the scientific work and the others for a broader communication of how science can help them.
Right, absolutely. And that is part of why we've built this repository, that through Scott's really great work, and trying to pull out the biological variation, which can often be very subtle, but still significant. And if you have technical noise in the way, that can really be problematic. I'll come back to that in a second, but I do want to touch on one thing you said about education, because that certainly is a piece of this, and just trying to spread broader awareness in the community that of the excitement of stem cell research, of the value of it. And we do a lot of this at NYSCF too. And recently I was speaking to some high school students, and they were very, very excited about it and all of that. But they, one of them asked me, and this has happened before too, like, what are the risks though? What are the risks for me of participating? How do I know that it's safe and that it's okay? And certainly they're not talking about, is this going to hurt me or something in some way? They're talking about, some of these reasons for mistrust. John, I'm curious as to your perspective. What do you say when people ask you things like that?
It's a very different relationship when you're dealing with a family in a clinic where they're seeking an answer for their kid with a rare disease versus talking to the Puerto Rican population in the Bronx. In the former case, it's relatively more straightforward to build trust. We go through an actual consenting process for doing any genetic testing. We talk about confidentiality. They're used to the idea that intimate information about their health is in our electronic health record. That's a foundation in a way, because these individual instances of trust can be knit together into a larger structure for a community as a whole, which is how a health system can I think do a lot more good than a research institution, because a lot of those bridges are already built. However, having said that, we are very aware of the fact that we have massive challenges if we're going to apply genotyping to help gain insights into disease risks, into adverse drug reactions, anything that might be valuable for our patients in the Bronx, because at that stage, we're not talking one-on-one to individuals who are highly invested in getting an answer. We're talking to community representatives, and community representatives are there to help make sure that there is no exploitation of their communities. So it's a different type of a conversation. I'm not going to say that we have all the answers, but it has to be participatory, and it has to be something where we use the trust that we've built as a health system to try to build a new bridge that will allow us to have a genomics offering for the communities of the Bronx, and not regarded as just one large group of people who are other. It has to be focused on specific communities. And I think as a final point, it should be focused on disease. Don't go out there and say, I'm going to sequence 400,000 people in the Bronx and we're going to get answers about everything. Go out there and say, what's important to you in the Bronx as a person who lives here? And if they say asthma, then we'll study asthma. We'll do it in a disease-focused way.
Yeah, absolutely. Ekemini, you had mentioned the Global Parkinson's Program, GP2. This is this big initiative that ASAP is engaged in to look at the diverse genetics of Parkinson's. I think a really promising example of the initiatives that are now underway. Can you tell us a little bit more about that? What you've been able to do, and what kinds of challenges you might still need to overcome?
Sure. So, as I mentioned, the Global Parkinson's Genetics Program is a worldwide program to genetically, no, let me backtrack. We are building the most ancestrally diverse genetic database for Parkinson's in the world. So the mission here is to dramatically expand our understanding of the genetic basis of Parkinson's disease, which is a global issue, and to make that knowledge globally relevant. So in doing so, we also hope to enable discoveries both in diverse populations, but also develop cellular models, create a framework for being able to examine gene and environment relationships. So really thinking about creating that rich foundational data set from which investigators can really pull from. And that's something that's missing in the Parkinson's disease field, as well as several others. And so we are on pace now to genotype over 150,000 ancestrally diverse participants. And I think one of the things that has really been able to fuel and drive that forward, as I mentioned before, is the intentional engagement with various communities. So within that, we have, while we are in all of the continents, we've also been able to sort of drill down on certain populations for which we know we need to sort of mobilize more support. So we've had the Black PD initiative that's embedded within GP2. So that's the Black and African American Connections to Parkinson's Disease study, as well as the large PD study, which is the Latin American Research Consortium on Genetics and Parkinson's Disease. So, again, doing this worldwide takes a lot of coordination. It also takes working with communities on the ground in place. So not necessarily, coming into every country and saying, hey, give us all your genetic material, but working with infrastructure that's in place. In many cases, building infrastructure with local universities to sort of bolster what they have and also provide what they may need to enable this work. So as I mentioned earlier, we can't just swoop in to different communities and take, right? So if we're going into Nigeria or South Africa or places in India, and we're working with their local universities, and they say, well, we need XYZ resource to be able to enable this, and also to enable our local physicians to be able to collect the material that's needed, then we know, okay, we need to bring the resources together to make that happen, right? And I think that's also something that we need to be mindful of, both as funders, but then also as investigators, is that we can't just go into communities and expect to get far more rich data without providing the resource to do so, right? And so I think what I've learned along this whole pipeline of standing up GP2, working with our two co-PIs, Dr. Andy Singleton and Dr. Cornelis Blauendrad, is that it is super important to engage the communities that you're working with everywhere. And this is everything from making sure you have materials in the languages that are relevant for that area, making sure you're actually bringing everyone on board with how data is being collected, how data is being disseminated, how it's being shared across borders. All of those discussions being had up front coming to agreement and really sort of enable the process to run that much smoother. So I think you asked about challenges. I think all of the things that we're talking about are things that we anticipated up front, things that we knew going in. We're saying, okay, we want to build the most ancestrally diverse resource for Parkinson's disease. What are all the issues that we're going to have to jump over? And all of the issues that we've discussed in this conversation are ones that we carefully thought through, which is why we've gone through the process that we've gone through in terms of going into various communities and really enabling work to happen in place. I think is probably, at least from my view, one of the biggest gains that I've seen with this program.
That's really excellent. Did you have any other similar initiatives to learn from as you were putting this together, or was there a lot of,
I think there's a lot of
lessons
learned from across the PIs in our steering committee, as well as the PIs that are leading this, who are, renowned geneticists, have done large studies over time, and sort of saying, what are the issues that we need to overcome, and how can we start from the beginning with this big initiative and try to mitigate those issues coming forward. Will there be new issues that arise? Certainly, but I think, as we know the issues that exist, all the things that we're talking about now, these are issues that have existed, right? I think what is happening now is that we're seeing the tide turn of people being willing to roll up their sleeves and actually deal with these issues, actually think about solutions to mitigate a lot of these issues, which has led us to where we are right now, which, I'm both ecstatic about and really proud of.
Well, that's really great, rather than just sidestepping them or trying to get around them. Right. Yeah, that seems to have been the sort of mode before, but that's really excellent work. And, I hope that you're able to share your lessons with others who are starting these kinds of initiatives, because it sounds like you've really, taken this on in a comprehensive way. And so that kind of brings me to the final topic I'd like to discuss with everyone in our last few minutes here is, kind of what we can all do to take action together. We clearly have technology available that allows us to take this on. We have momentum here in terms of actually confronting these issues rather than sidestepping them. But we don't quite fully have the social and cultural competency and the resources that we need to achieve a really inclusive disease research. So this is a really huge undertaking, but definitely somewhere where we're, we can all make an impact and a difference. So in terms of, setting up collaborations and that sort of a thing, Ekemini, you and I have talked about how this does need to be a really collaborative effort. No single one of us here is going to be able to solve this, alone. Let's talk about the funding aspect of it, because clearly there's a resourcing issue. We have the will, we need sort of the way. How do you think about this from a funding perspective, and what do you think it'll take to bring funders on board? Ekemini, if you want to start, and then
I'll kick us off. I think,
as our resident funder,
I think there needs to be a willingness to have strategic partnerships. And I think a lot of times in science we talk about egos of investigators, but I think there's egos everywhere, including egos with different organizations. Everyone wants their name stamped on something, and I think in favor of solutions, we should be, as funders, very willing to align and go at different problems together, both cost sharing, recognition sharing, all of that, I think, is something that we need to be more willing to do, and more willing to put the dollars behind where we say we find importance, right? And so, at some point, it'd be fantastic. Like, let's have a collaboration between us and Allen Institute. We have collaborations across the board with various organizations, and so we are working with NYSCF. So I think there are more and more funder collaboratives, more and more joint funder efforts that need to be supercharged and with resources and funding jointly combined to enable this kind of work.
Absolutely. And Ru, we've talked a little bit about your efforts in the realm of appropriations. Could you tell us a little bit more about that?
Yes, so I think this kind of work that all of you amazing panelists have spoken about takes time as well, right? That time is also a resource, I guess. and learning from each other, because as speaking for cell science, our Institute for Cell Science, we can go and obtain the samples, do all the right consent. We don't have the expertise in that, or even to generate the lines like Scott has done, right? So I think partnering and collaborating is absolutely the key. The second part is that even just doing, it's always a side project. These are not things that some of us haven't thought about before. It's just always been something on the side. So I think making this a priority, and that's why the resourcing is so important, it has to be a priority. Otherwise, it just won't get done. So those are the things we're tackling with. There are other legal, IP, all of these issues too that come along with generating a cell line, for example, and sharing with the public, which we had to do upfront, even to use the cell line we use now to do our research. So currently we are just talking about all of this, but specifically to the appropriation, I think having the government and the granting agencies also know that this is something that's important all the way from the top, helps fund different groups. Because again, even if 3 institutes do it, it's not enough. If we can get more people on board and give them the funding to make this a priority, I think that's when we'll really see the tide turning.
Absolutely. Scott, as you talk about, oh, John, did you want to add something?
Defer to Scott, but I'd like to add something after.
Okay, well, I was just going to say, you know, we're working on some of this stuff too. What are your thoughts on how to bring in the right collaborators and funders to align on these issues?
Yeah, I think, you know, working with disease foundations, for example, you know, being able to, you know, collaborate and cooperate in those, you know, through those foundations that have built trust with their patient populations already is one sort of avenue. But one of the things that I would love to see from the funding perspective is a vision for ongoing support for maintaining those relationships in the community, and those, you know, whether it's through the disease foundations, whether it's through community support. But, you know, as we turn our thinking from just a one-time go collect a sample and we're done and that's it, turning our thinking into this ongoing sort of communication and you know, back and forth that's going to be necessary to realize all this, having the support for that sort of long-term maintenance of those relationships. You know, there's a cost to doing that, and that's something that we need to sort of think upfront about how to build that into the programs that we're creating.
Yeah, absolutely. And the same thing goes for infrastructure. There isn't really a whole lot of funding out there for infrastructure. So when, you know, we were all working together, Scott and John, on, you know, building a diverse biobank and a sort of targeted piece of that, it was pretty difficult to find a funding call that would fit that. And that is something that is definitely needed out there. John, maybe you were going to talk about this, but yeah, what were you going to say?
I think the experience of COVID was very interesting in New York City. People really mobilized to try to understand the variants that were present in the New York population. But a funny thing happened. About 80% of the sequencing that was done was on people who were attending Manhattan clinics, where the death rate was about half what it was in the Bronx. So when we started to talk more broadly on a mayoral task force about how to implement a surveillance system during COVID in 2021, it became apparent that what we were doing, these very well-intentioned people who were working really hard to do a lot of genotyping in New York City, were in fact exacerbating health disparities, because they were sequencing the people who didn't need it as much as the people out in Queens, Brooklyn, and the Bronx, where we had the highest per capita death rate of all. So this is an issue that I call research disparities. We know that there are maps for health disparities where we can talk about access to services and so on. We don't have maps for research disparities. And what you'll find is that people who make these big biobanks in their own institution are generally doing it with donor money, where there's a lot of philanthropy, and they, by definition, are places which have wealthier groups of people around them, and probably not communities of color, probably not minoritized communities. So we have to have a way of recognizing that as a problem, first of all, and then redistributing funds in a way that we are starting to bring the funds to the communities, where we can then talk about having inclusiveness as a result, because research disparities are real, and they're contributing exactly to this issue that we're talking about today.
Absolutely. Well, in our last few minutes here, I want to ask each of you for a closing thought on how those in our audience, be they scientists or otherwise, what can they do to help? Ekemini, do you want to start?
Oh, I'm trying to think of which perspective I want
to. I know you bring a lot of lenses.
Yeah. I guess from, I'll speak about this from the sort of scientists at the bench perspective. And I think, as you're choosing your cell lines for your studies, make the extra effort to choose cell lines that are diverse in gender. I mean, there are tons of male cell lines, but incorporate a female cell line, and incorporate a cell line from a different ethnicity. It's hard for many of the diseases that we study because they simply don't exist yet. But where they do exist, I think make that extra effort to incorporate those cell lines.
Great point. Ru, would you like to go next?
Yeah, I want to second Ekemini there. I think when there's a demand, the demand gets met somehow. So I think always thinking about the biology you are doing and what is it that you're trying to understand, and are you being constrained by the cells you use, or the sample you use, or the tissue you use, right? I mean, having that awareness, because 50 years ago, we didn't have that luxury. We had to use what was out there. I think with the technologies available today, it's more up to the research. And of course, if there's some preliminary data from those, it'll lead to other studies. I think it'll lead to more funding, right? If the funders see this is actually doable. So that's kind of my main thing that I would like to leave you with.
Thank you, Ru. Scott?
Yeah, I think just sort of stepping back and shifting our thinking from having this whole issue be just a side project that we want to shift to thinking about integrating it and actively pursuing, ways of trying to correct the issue. So being more active and purposeful in the way that we approach the way that we design experiments, or the way that we design research studies. Being a little bit more mindful and more active and purposeful in the way that we design the work that we do. I think that would be my hope that people would take away is to shift your thinking, to be more purposeful, to try to correct the problem.
Yeah, right from the get-go, not at the end when you need to
Build
tick a box on. Yeah, absolutely. Great point. John.
I think we're going to be encouraging people to step up and to engage in this conversation. So given that, I'm going to use my time to answer the question in the Q&A, because if Andrew Gaffney has raised a nice question, he should have the courtesy of having one of us respond. So Andrew, you've raised the issue about whether European, African, South Asian, and East Asian ethnic ancestral groups are enough to be able to capture variation. Clearly not. There's so much more that we could and should be doing. I'm very interested in all of these little subgroups I have in the Bronx, but I recognize that there are places like the Indian subcontinent where there's so much to be discovered. There is basically an incredible world of variation out there. So the question is, how do you prioritize? And I think that the simple thing to do, because it's a deliverable for your community that you're engaging with, is to engage by disease. So you don't have to necessarily get all of the major ancestral groups. You don't even necessarily have to be inclusive. What you want to do is to say, there's a disease. I know that it has a particular prevalence, or it's causing harm in this community here. Let's focus on that. Let's talk to them. Let's try to figure out how to address that disease with that community. And then you've captured diversity by looking at that community and comparing it with unaffected groups. That probably is the more effective way of addressing a lot of the issues that we have raised in this really interesting discussion today.
Thank you so much, John. Great point. And I would also say to those of you who are not actively practicing science, if you're interested in ways that you can get involved, certainly encourage you to use all of the advocacy channels you have available to you to advocate for funding and resources to be able to support this research. As I said, we have the technology and the momentum. We really just need the resources to get it fully in place. So we really appreciate that, and we'll share some links afterwards and how you can get involved in NYSCF's research if that's of interest. So we're out of time. Unfortunately, I could have spent the entire day or entire week with all of you. I'm so grateful to you all for being here for this really important discussion. This is certainly not the end of it at all, but a continuation on this really important topic. So thank you so much to our fantastic panelists for all the work you do every day and for this great discussion today. Thank you all in the audience for your participation. And I apologize if we didn't get to everyone's questions, but we will follow up as far as possible with that. So hope to see you all at a future NYSCF event, and thank you all, and stay safe, be well.